Welcome to Bioratna. Dear students, today we are going to discuss the structure of DNA. DNA was discovered by Friedrich Mischer. It was discovered by Friedrich Mischer in the year 1869. He found it in the nucleus of pus cells, and he named it as nuclein. Now, Watson and Crick, they proposed the structure of DNA in the year 1953. This is a long time gap, 1869 to 1953. But during this period, many work was going on to find out the biochemical nature of DNA as well as the structure of DNA, which helped Watson and Crick a lot in finding out the structure of DNA. Let us see one by one. Okay, so it was Albrecht Kossel in the year 1881. He found out two things. One, that DNA is this nuclein of Mischer is acidic in nature, and second thing is that DNA is made up of 4 types of nitrogen bases: A, T, C and G. Okay. Next comes Richard Altmann. Richard Altmann in the year 1889 names DNA as this nuclein as nucleic acid. Okay. Next is Ashbury. It is a very important finding. This is in 1940s when Ashbury discovers that DNA is actually a polynucleotide. DNA is a polynucleotide. Means nucleotides are the basic units of DNA, and when many nucleotides they join together, then they form the deoxyribonucleic acid, that is the DNA. Now students, there is 2 line of investigation which helped Watson and Crick a lot, and these 2 is, one is by Chargaff and the another is Wilkins and Franklin. Let us see what it is. It was Chargaff in the year 1950. He states that in a DNA molecule, the number of adenine A always equals the number of thymine, that is T, and so A by T is always 1. Then the number of guanine also equals the number of cytosine, so G by C is equal to 1. Great finding. Now, next is Wilkins and Franklin. This is the same year when Watson and Crick found out the structure of DNA, that is this 1958. In 1953. In 1953, Wilkins and Franklin, by their X-ray crystallography से उन्हें पता चला कि DNA is a double helix. So they found out by DNA X-ray crystallography that DNA is a double helix. Okay, so they found out that DNA consists of a double helix. These many findings were available to Watson and Crick. Now we see next as we know that DNA is a polynucleotide, we have to understand what is a nucleotide, what is the structure of nucleotide. Then by joining together of the nucleotides, we will form one strand of DNA. Then forming complementary strand, we will see how to make the second strand of DNA. So our next subtopic is the structure of nucleotide. Now we see what is a nucleotide. As you already know that nucleotide is the monomer of DNA. ये एक सिंगल मोती है जिसके जोड़ने से आपकी एक मोती की माला बनने वाली है, that is called the DNA. So nucleotide is the monomer and many nucleotides, they join together to form the DNA. And this nucleotide, it has three components. What the three components? One is deoxyribose sugar. Deoxyribose sugar. Another, this is nitrogen base, and the third, the phosphate group. When these three join together, then the nucleotide will be formed. Let us see what is deoxyribose sugar. Ribose sugar is let us first see ribose sugar. The ribose sugar is a pentose sugar. Pentose means having 5 carbon. So it is a pentose sugar found in RNA where 4 carbon and oxygen, 4 carbon and 1 oxygen, they form a ring. Where are the 4 carbons are there? 1 carbon here, another at this point, another at this point, and another at this point. Now I am explaining. So here for the first time for beginning, now I have written mentioned the carbon here. Afterwards you have to understand, you have to know that at each of these corners, means if this is oxygen and if it is drawn like this, then at every point there is 1 carbon. So these 4 carbon, this is C1, this is C2, this is C3, this is C4. 4 carbon and this oxygen, they form a complete ring. Then where is the 5th carbon? The 5th carbon is outside. The 5th carbon, it is outside. Here it is CH2OH. Now to this first carbon, here it is attached, here it is OH, this is H. This is H, OH. This is, we are seeing the ribose sugar. So here it is OH. Now here also C3H, OH. Here H. This ribose sugar, all of you know the empirical formula of carbohydrates. Carbohydrates का formula होता है CnH2ON. The empirical formula of carbohydrate is CnH2ON. So here the N is 5, so carbon is 5. Hydrogen twice the number, that is 10. Oxygen same, that is 5. So this is C5H10O5. This is called ribose sugar, and this ribose sugar, when from this C2 position, from here, when 1 oxygen is missing, this oxygen is not there, then it is called deoxyribose sugar. This is ribose sugar present in RNA, and here we will see deoxyribose sugar. The same thing, this is the oxygen. Now the oxygen forms a ring with 4 carbon. So here the 4 carbons are there. Now no longer we are going to mention carbon anymore. So now this is the it. Here it is CH2OH. one OH group here, here H, here also H. In C2 position, one OH, which is a very reactive group, is missing. At C3 position, one OH, here one H. So you see, in ribose sugar, there are 1, 2, 3 and 4 OH group, which makes RNA a very reactive molecule, whereas in DNA, this deoxyribose sugar is present. This is deoxyribose sugar, in which the oxygen is missing from this position. This makes it less reactive. This OH at C1 position, what is its role? It is there, it will react with the, आपको पता है कि अभी 3 लोगों को जुड़ना है, the sugar, the nitrogen base, the phosphate group. This is the sugar. With this C1 arm, यहाँ पे वो नाइट्रोजन बेस के साथ जुड़ने वाला है. यहाँ पे वो नाइट्रोजन बेस के साथ जुड़ने वाला है. एक OH गया. ये 5 वाला जो है, this OH, here it is going to join with the phosphate group. 2 से जब वो जुड़ जाएगा, then nucleotide will be formed. What about this OH at the C3 position? When these 3 means this sugar, this nitrogen base and one phosphate group get attached, then only one nucleotide will be formed. बहुत सारे न्यूक्लियोटाइड्स को जुड़ना है, तभी जाके डीएनए बनेगा. सो इस OH की मदद से वो दूसरे न्यूक्लियोटाइड से जुड़ता है. It gets hold of another nucleotide with the help of this OH group, which is present at C3 position. Okay, so this is about the sugar. We will see now what is nitrogen base. Before that, one revision. Ribose sugar is a pentose sugar. Pentose means having 5 carbon atoms. It is present in RNA, and when one oxygen is missing from C2 position, then we call it deoxyribose sugar, which is present in DNA. Now we see the nitrogen bases. Okay. The nitrogen, the second component, that is nitrogen bases. The nitrogen bases in DNA, they are of 2 types. One is purine, another is pyrimidine. This purine, it has dicyclic nature, means 2 ring-like structures joined to each other, whereas pyrimidine is monocyclic in nature. This purine, it has 9 members. It is 9-member dicyclic molecule, whereas this one has 6 members. It will be clear from the skeleton which I am showing for the purine and pyrimidine. See, for purine, there are, this is a 6-member ring, and then to it is attached a 5-member ring. So together they become 9-member ring. In case of pyrimidine, this 5-member ring is missing. So it is not this. This will form a pentose one. Okay. So here 1, 2, 3, 4, 5 and 6. This is counted anti-clockwise, this is clockwise. This is the skeletal ring structure for purine and pyrimidine. In DNA, this purine is of two types. One is adenine and the other is guanine. Adenine and guanine. Whereas the pyrimidine also is of two types, that is cytosine and thymine. Cytosine and thymine. So, purine is of two types, A and G. Pyrimidine is of two types, C and T. We have now seen sugar. We have also seen about the nitrogen bases. Now the phosphate group. The phosphate group is H3PO4. It is phosphoric acid. H3PO4. This is P, one OH, another OH, another OH. कितने हो गए? P. This is 1, 2, 3 oxygen. So one more oxygen needs to be there. 1, 2, 3 hydrogen. This H3 we have written, P written, one more oxygen. Here one more oxygen with double bond. Now when these three go on joining together, then the nucleotide will be formed. Let us see. Now when a sugar molecule joins with nitrogen base, their three molecules need to be joined. Here first we are seeing only two molecules. When sugar is joined to nitrogen base, then what is formed? Then not nucleotide, one step before that, this is nucleoside. When sugar joins with nitrogen base, then nucleoside is formed. This nucleoside is of four types because we have four types of nitrogen bases. अगर ये nitrogen base हम लेते हैं adenine, then this nucleoside is adenosine. If it is guanine, then this is guanosine. If it is thymine, then this is thymidine. Here it was SINE, now it is DINE. Similarly, for cytosine, it becomes cytidine. You think that this is hard to remember? Not at all. It has a rhythm in itself. Like the nursery students, they say the poems in a rhythm. This automatically comes. I have never found any student having any problem remembering all the nucleoside and nucleotide names. This is adenosine, guanosine, thymidine, cytidine. So this is nucleoside. मतलब ये आपने जो शुगर बनाया था, यहाँ तुरंत आप ऐसे लाइन लगा देते हो, यहाँ पे CH2H जैसे का वैसा है, यहाँ वन पोजीशन में एक नाइट्रोजन बेस लग गया है. सो यहाँ पे एक नाइट्रोजन बेस लग गया है. सो दिस इज द नाइट्रोजन बेस हियर. सो दिस इज nucleoside. When here at one position nitrogen base is attached, then you call it as nucleoside. अब चलिए बनाते हैं इससे nucleotide. Now here this is nucleoside. When it attaches to a phosphate group, then we get the nucleotide. So if when adenosine attaches to phosphate, then it is adenylic acid. Phosphate group is acidic, phosphoric acid. So now it will become acidic. So now the name is adenosine changes to adenylic acid. Guanosine becomes guanylic acid. Thymidine becomes thymidyllic acid. And cytidine becomes cytidyllic acid. The unit is made. Here this diagram we will see, this diagram we will draw. This one, what we had done here, this oxygen, this 5 carbons, 1 carbon outside. Here it has already joined to the nitrogen base, and now here at the 5th carbon, it will attach to the phosphate group. This bond, here the sugar and nitrogen base, they are joined. This is called N-glycosidic bond. N for nitrogen, glycosidic for this sugar. So this bond is called N-glycosidic bond. This is N-glycosidic bond, and this phosphate और ये शुगर के बीच का जो ये बॉन्ड है, फॉस्फेट और शुगर के बीच का जो बॉन्ड है, this is called ester bond, phospho-ester bond. This is phospho-ester bond. Now, just from this diagram, we will see what is a nucleoside and a nucleotide. A nucleoside, when to the sugar molecule, nitrogen base is attached in one position, then we call it nucleoside, and when to its 5th position phosphate is attached, then we call it nucleotide. The bond between nitrogen base and sugar is called N for nitrogen base, N, and glycosidic sugar, N-glycosidic bond, and the one between this phosphate and the sugar is called phospho-ester bond. So the monomer is ready. We will now see next that by joining these monomers, how the polymer DNA is formed. First we will make one DNA strand, and from that we will see how both strands are formed.